Welcome, everyone. We all know that clinical research is built on details. That is a fact. And right at the center of all those details is the clinical trial protocol. Now, if you've ever opened one, you know, it's not just a research plan, it's a contract, a playbook, a rule book, and in many ways, it's the heartbeat of the entire study. In today's tutorial, we're going to take a closer look at the structure and purpose of a clinical protocol. You'll learn what goes into it, why every section matters, and how it's used by everyone, from investigators and sponsors to regulatory bodies and monitors. Then we'll move into how protocols evolve, because trials don't stand still, and neither do protocols. We'll walk through how protocol amendments are handled, when they're necessary, and what rules apply before you can make those changes. We'll also address a source of confusion in almost every study team. What's the difference between a protocol deviation and a protocol violation? We'll clarify those definitions, give real examples, and help you understand the consequences and how to manage them properly. Finally, we'll talk about the regulatory side of things, because writing a protocol is only half the job. Submitting it to the right authorities, getting the right approvals, and tracking any amendments is a key part of doing research that's ethical, compliant, and trusted. So whether you're just starting out in clinical research or you've been working with protocols for years, this session is going to give you the clarity and context you need to apply all of this confidently in your day-to-day work. Ready to dive in? Let's go. As I mentioned earlier, the clinical trial protocol is central to every clinical trial, yet often misunderstood or taken for granted. The clinical trial protocol. You've probably seen one before, maybe even flipped through its pages. It's that long, detailed document filled with objectives, timelines, eligibility criteria, endpoints. But what is a protocol really? Well, think of it this way. A protocol is the blueprint of a clinical trial. It's not just paperwork, it's the master plan that tells everyone involved what to do, when to do it, how to do it, and most importantly, why they're doing it. It defines why the study exists, how it will be conducted, and what success looks like. And without a solid protocol, you don't have a trial, you just have chaos. So what's the purpose of a clinical protocol? The purpose is threefold. Scientific clarity. It lays out the research question, the hypothesis, and the rationale behind the study design. Regulatory compliance. It ensures the trial meets ethical and legal standards aligned with GCP and regulatory bodies. Operational consistency. It keeps everyone, investigators, study coordinators, monitors, sponsors, on the same page. A well-written protocol reduces ambiguity, supports safety, and helps ensure that the results will actually be interpretable and credible. Now let's break down the main components of the protocol structure. Section by section, what's inside a clinical trial protocol? Here's what you'll typically find. 1. Title page and protocol ID. It includes the protocol number, study title, sponsor, version, and date. This is critical for traceability and version control. 2. Table of contents. Not just a formality, this helps users navigate a lengthy document quickly and accurately. 3. Background and rationale. Why this trial? Why now? This section justifies the study's need, summarizing existing data, gaps in evidence, and the scientific reasoning. 4th, objectives and endpoints. This section clearly defines the primary objective, e.g., proving efficacy, secondary objectives, e.g., quality of life, safety, and the clinical endpoints used to measure success. This is what the whole trial is aiming to achieve. 5. Study design. Is it randomized, blinded, placebo-controlled, parallel group? This section gives a detailed layout of how the trial is structured and how participants are assigned. Sixth, subject eligibility criteria. Who can join and who can't? Inclusion and exclusion criteria ensure that the right population is studied and that patient safety is maintained. 7. Treatment plan. What interventions will be given, at what dose, and on what schedule? This part maps out how the investigational product or placebo is administered. 8. Assessments and procedures. How and when will data be collected? Think lab tests, imaging, physical exams, questionnaires. This is the how of measuring outcomes. 9. Safety monitoring and reporting. This section outlines how adverse events will be identified, recorded, and reported, ensuring participant safety throughout. 10th, statistical analysis plan. This explains how the data will be analyzed, what statistical methods will be used, how missing data will be handled, and what sample size is needed. 11. Ethics and regulatory considerations. Here you'll find references to GCP, IRB approval, informed consent processes, and handling of protocol deviations. 12th, data management and record retention. Where will the data live? How will it be stored and protected? This section ensures compliance and transparency. 13. References, appendices, and signature pages. These are often overlooked, but they're critical for documentation, investigator agreement, and protocol approval. As you see, this is important because whether you're in medical affairs supporting investigator-initiated studies, in clinical operations managing trial logistics, or an investigator enrolling patients, the protocol is your roadmap. It guides your actions, it protects your participants, and it ensures your study can pass regulatory scrutiny and generate meaningful results. A poorly written protocol creates delays, confusion, protocol deviations, and in some cases, puts patients at risk or invalidates data. A well-written protocol? It builds credibility, supports compliance, and improves the quality and reliability of your trial outcomes. Now let's talk about handling protocol amendments. Protocols are supposed to be well-planned, carefully written documents, but clinical research rarely follows a perfect straight line. Trials can span months or even years. During that time, science evolves, safety signals appear, recruitment patterns change, and even new regulatory requirements may arise. So what happens when you need to change the plan? That's where protocol amendments come in. A protocol amendment is a systematic change or formal clarification to the original protocol. This could be something as minor as fixing a typo, or as major as changing the primary endpoint, adjusting the sample size, or modifying the eligibility criteria. Now here's the important part. You cannot just make changes and move on. Every amendment must go through a structured approval process to maintain compliance and protect patients. There are generally two types of protocol amendments: substantial or major amendments. These impact participant safety, scientific validity, or how the trial is conducted. For example, adding a new study arm, widening or narrowing inclusion/exclusion criteria, changing dosing frequency, updating safety monitoring procedures, non-substantial or minor amendments. These include administrative corrections or clarifications that don't impact trial conduct or participant safety. For example, typo corrections, clarification of wording, update of contact information. So then, how to handle protocol amendments? Here's the standard process. 7 important steps, 1 through 7. Draft the amendment in collaboration with relevant teams: medical, regulatory, clinical operations, and biostats. 2. Review internally to assess the scientific and operational impact. 3. Submit to the IRB/IEC for review and approval, especially for substantial changes. Submit to regulatory authorities, e.g. FDA, EMA, where required. 5. Communicate the amendment to all investigators and relevant parties, clearly outlining what has changed and how it affects the study. 6. Train staff on the updates and document everything. 7. Implement only after approvals are in place, never before. And don't forget, every amendment must be dated, version controlled, and stored with the essential documents. It also needs to be reflected in the clinical trial registry, if applicable. Poorly managed amendments can lead to protocol confusion, compliance risks, or worse, regulatory findings during audits. Now, let's move to a topic that causes a lot of confusion, even among seasoned research professionals. What's the difference between a protocol deviation and a protocol violation? These terms are often used interchangeably, but they shouldn't be. Starting with protocol deviation. A protocol deviation is a departure from the approved protocol that is unplanned and typically minor. Examples include a missed lab visit, slightly delayed dosing, incomplete questionnaires, a study procedure performed outside the scheduled window. These often occur unintentionally, and while they must be documented, they may not always impact subject safety or data integrity. Still, too many deviations can indicate systemic issues at the site and may trigger a compliance investigation. What about protocol violation? A protocol violation, on the other hand, is a serious breach, one that could impact participant rights or safety, the validity and reliability of trial data, compliance with GCP and regulatory requirements. Examples include enrolling an ineligible participant, failing to obtain proper informed consent, administering the wrong dose or wrong investigational product, failing to report a serious adverse event, SAE. Violations are reportable to the sponsor and sometimes to regulatory authorities. They may lead to subject withdrawal, site retraining, or even disqualification of data from analysis. Now let's break it down side by side. Protocol deviation versus protocol violation, so the differences are clear and memorable. Look at the first row of this table. Severity. Protocol deviations are usually minor, things like a slightly delayed visit or a form that wasn't filled out completely. But violations? These are serious. They can compromise safety, data integrity, or even the ethical foundation of the study. Next, impact. A single deviation might not seem like a big deal, but if they start piling up, they can point to bigger issues at the site. Violations, however, almost always carry significant consequences. Think, invalidating subject data or putting participants at real risk. Reporting requirements come next. Deviations need to be documented. That's non-negotiable. But violations? These usually must be reported immediately to the sponsor, and in many cases to regulatory authorities or ethics committees as well. And then we get to examples. A protocol deviation might be something like conducting a lab test outside the scheduled window, or missing a non-critical questionnaire. But a protocol violation? That's when you enroll someone who doesn't meet inclusion criteria, fail to obtain proper consent, or administer the wrong investigational product. These aren't just mistakes, they're breaches of GCP. So here's the takeaway. Not every error is a violation, but every departure from protocol matters. Understanding the difference helps you respond appropriately, prevent recurrence, and protect both your trial and your patients. Understanding the difference isn't just a technicality, it's a core GCP competency. For investigators, it helps you take corrective action, avoid repeat issues, and maintain your site's credibility. For medical affairs and clinical operations, it allows you to support sites better, monitor trends, and prevent future risks. And for sponsors, it's essential for evaluating site performance, triggering CAPAs, corrective and preventive actions, and responding confidently during inspections. Let's talk about a part of clinical research that often makes people pause: regulatory submissions. It sounds formal, it sounds technical, and yes, it is. But it's also one of the most essential parts of running a trial the right way. Because no matter how brilliant your protocol is, if it hasn't been reviewed, submitted, and approved by the right authorities, you're not ready to start. So, where does it begin? With the original protocol, the foundation of your study. Before a single patient is enrolled, this document must be submitted to your local ethics committee or institutional review board, IRB, IEC, and to the relevant regulatory authority, like the FDA in the US, EMA in Europe, or local ministries of health in other countries. This submission typically includes the full protocol document, the informed consent form, investigators' brochure safety information, any other supporting materials relevant to patient safety or trial procedures. These bodies aren't just rubber stamping your paperwork. They're reviewing for ethical soundness, scientific merit, and patient protection. If they find issues, they'll send back questions or request modifications. And no trial activity can begin until approval is granted. Now let's talk about amendments. Protocols are living documents. Things change. New data becomes available, eligibility criteria may shift, safety signals may arise, or logistical adjustments might be needed. That's where protocol amendments come in. But here's the key. You can't just implement a change and notify regulators later. Every substantial amendment, meaning anything that could affect participant safety, study objectives, or data integrity, must be submitted for review and reapproval before it's put into practice. This applies to new arms being added, changes in dosing, alterations to inclusion/exclusion criteria, new investigational sites or investigators, updates to the informed consent form. Even smaller tweaks, like clarifying ambiguous text, might still require submission depending on local laws or the study's risk level. Why does this matter so much? Because regulatory compliance is not just about bureaucracy. It's about transparency. It's about documenting intent. And it ensures everyone, regulators, ethics committees, investigators, and participants, are aligned on what's happening in the trial. Skipping or delaying submissions, that can lead to audit findings, trial holds, or even invalidation of your data. A quick word on timelines. Different countries have different expectations. Some require pre-approval of changes before they're implemented. Others may allow notification-only amendments, but only for non-substantial changes. That's why your regulatory affairs team plays such a vital role. They interpret local laws, liaise with authorities, and ensure that every version of the protocol submitted and approved is the one that's being followed on the ground. So whether you're designing a new protocol or updating an existing one, remember, every step in clinical research must be intentional, documented, and approved. It's not about slowing down innovation. It's about doing it ethically, safely, and with full accountability. And there you have it. We've covered a lot in this tutorial because protocols are no small matter. We started by exploring the structure and purpose of clinical trial protocols, how they bring clarity, consistency, and accountability to every phase of a study. Then, we looked at protocol amendments and how to handle changes properly, with the right approvals and documentation. Because research evolves, but we still need to stay within the guardrails. We also drew the line between protocol deviations and violations, a distinction that can protect your trial's credibility and your team's compliance record when understood clearly. And finally, we took a practical look at regulatory requirements, because what matters on paper only matters if it's been reviewed, approved, and aligned with the rules. At the end of the day, protocols aren't just technical documents. They are ethical tools. They are accountability frameworks. And they are what make clinical research trustworthy, both for regulators and, most importantly, for the patients who agree to be part of it. Thanks for being here, and see you in the next session.